Yeah, perfect. So my talk today would focus on designing a multi-responsive polymeric amphiphiles with high molecular precision. And I think also in light of having this conference in memory of Francois Winnick, I decided to share with you very often when we think on multi-responsive materials, we can think on materials that can respond to different types of stimuli. And what you can see here is a 3D printed octopus that was printed with layers of poly and isopropyl acrylamide, and therefore when you change temperature, these legs can fold or refold. Or it has also some acrylic acid monomers, so it can swell if you take it into a basic pH. But if you think on multi-responsive materials, you can also think on materials that respond in a couple of different ways to the same stimuli, and this would be more of the focus of my talk, on materials that can both disassemble in response to a stimuli, so mostly polymeric micelles, but they can also change their spectral properties due to the same response. So my group is mostly interested in the use of enzyme as a stimuli, and we heard a great talk this morning about enzymatic degradation of polyesters. And the reason we are very interested in studying enzymes is the fact that enzymes are highly selective. We all know that they have catalytic capabilities. They are already present in the body. And most importantly, we are more and more aware of specific enzymes that are expressed in specific diseases or specific tissues. And this means that if we can learn how to design polymeric carriers that would respond to this specific disease-associated enzymes, such as you can see for here an example of catepsin, overexpression of catepsin B, and it's not only overexpressed inside the breast cancer tumor cells, it's also being excreted to the extracellular matrix. So if we could learn how to design material that can respond to such disease-associated enzymes, we should be able to achieve a very selective type of therapy. So in order to study the ability of enzyme to degrade polymeric materials, we went for a very unique architecture, which is a hybridization between a linear polymer and a dendrimer. And the idea is that the linear chain would be hydrophilic. The hydrophobic part would come from the dendron, and more specifically from the hydrophobic end groups that decorate the dendron, and they would be linked to the dendritic branching architecture through enzymatically cleavable bond. And the reason we went for this architecture is that it should give us two main advantages. First, now all cleavable end groups are exactly symmetrical to each other due to the high symmetry of the dendron, but they are also now all terminal groups. So they all sit in the terminal of the branches and should allow a better or easier access of the enzyme to each of these hydrophobic chains and the ability to cleave them, more than if they would be located along a certain polymeric backbone. So we were definitely not the first one to think on this architecture, and the work and this architecture has been inspired by the work of pioneering work of Jean Fréchet, Craig Oker, Ivan Gitzel, and Karen Woolley, and later different scientists around the world. You can see here my pickup of my polymer heroes using this type of linear polymer dendrimer hybrids. And in all of these works, you can really appreciate the high molecular precision that you can combine into the relatively easier preparation of polymeric materials. So over the last in 7 or 8 years, we really took this platform and used it in a very modular manner by changing different molecular parameters, such as the length of the polyethylene glycol chain, changing the hydrophobicity by changing the number of the type of end groups, changing the overall molecular weight, and very recently also changing the architecture of the hydrophilic block. And what I would like to first share with you is again, is the appreciation of what you can see with high molecular precision when you have such a well-defined block, which is the enzyme responsive block. And this would be the work of Rav Segal, published a few years ago. And when we initiated this work, we wanted to make sure that we'll design two enzymatic substrates that would be slightly different from each other, but we wanted to make sure that the enzyme can cleave both of them. So we made what we call this G0. So basically, we don't have a dendrimer at all. We have the hydrophilic polyethylene glycol chain, which is the 5 kilodalton, and we have this ester bond right here with either hexanoic or hexanoate or nonanoate chains, and we study them at a concentration that is way below their CMC in order to take away the consideration of whether we have micelles or not. And what you can really very easily see here is that if you ask the enzyme what it prefers to cleave, it prefers to cleave the longer aliphatic chain, the nanoate ester, and hydrolyze them to give back a hydroxyl group here. So we then took our dendritic architecture. We made the dendron that have 4 hydroxyl end groups, and we decorated it with a mixture, with a 1-to-1 mixture of the hexanoic and nonanoic acid. And we are expecting to get a statistical mixture of hybrids that contain dendrons that have 4 hexyl end groups, 3 and 1, 2 and 2, 3 and 1 hexyl, 3 and nonat, and all the way to 4 nonyl end groups. And what I want to show in the next slide is actually something that is not very common in the field of amphiphilic block of polymer, is the fact that, again, due to the high molecular precision, we can actually run HPLC and use a reverse phase HPLC to characterize our polymer and characterize the degradation. And what you can see here is that initially we indeed getting a baseline separation between the different amphiphiles that are, I want to remind you, they're different from each other by 3 methylene units, whether you have, again, the number of hexyl and non-H chains. And you can see here, you can see here the pure hexyl made with just hexanoate or the pure nonyl, and we can see it in the mixture. We're really seeing all 5 expected peaks. And then when we add the enzyme, we can see that the peaks that contain that are richer in hexyl are getting degraded first, and they all give eventually the same hydrolyzed polymer. And what amazed us is because of this baseline separation, we could actually now plot the specific concentration of each of these molecular species in solution. And what's really amazed us is the fact that each one of them had a very different degradation rate. Now, again, if you consider the fact that we are talking about polymers in a molecular weight of around 6.5 kilodalton that are different from each other by 36, or actually the protons around the 44 mass unit, it's actually an amazing response and showing how delicate this enzymatic degradation of polymeric amphiphile is to very small changes in the hydrophobic block. And I think that this work is also demonstrating a very, or is emphasizing a very challenging thing in the world of polymer therapeutics. And this is that very often we like to take a hydrophilic polymer and to use it to attach hydrophobic drugs, to use this as a polymer carrier. But the problem is that we cannot control the exact amount of drugs and where they're located on the polymer. And we can obviously not decorate all hydrophilic end groups with a drug because then the polymer would become insoluble. So we usually, I would say, people are decorating roughly 5 to 10% on average of the polymer. But because this is an average, it means that some polymer would have a very small amount of drugs on them, and other polymers might have a very large number of drugs on them. Now, if you throw this into water, which eventually all biomedical applications start with introducing your polymer into an aqueous solution, then you'll have self-assembly. And it's the polymers that contain only few drugs can probably exchange faster with the micellar structure and get degraded by the enzyme, when the one that their degree of hydrophobicity is a bit higher might not degrade and release the drugs at all. And this has actually been a major limitation and a reason for failure of many products or polymers in clinical trials, because they're not releasing enough drugs as expected based on simply their loading, their average loading capacity. So in the next part of the talk, I would like to focus on how can we study this very unique polymeric amphiphile in a more complex biological environment. Eventually, again, we want to go into their usage as nanocarriers. And in order to do so, you can obviously not use just dynamic light scattering or directly the HPLC, and we wanted to introduce some type of labeling that would allow us to track these amphiphiles. And moreover, it would allow the amphiphile to respond spectrally responds also to the enzymatic degradation. So the mechanism works the following. We'll introduce the labeling moiety by introducing a linker exactly between the hydrophilic and hydrophobic part. Based on the specific dye that we'll choose, the polymer would have some spectral properties. Then we'll put the polymers, the amphiphiles, into water. They would self-assemble into micelles. And now in the micellar state, these dyes are going to be in very close proximity to one each other. And this means that they will be able to interact with the neighboring dye. And depending on the type of dye or the type of mixtures, we can gain different what we call supramolecular spectral properties. We can have dyes that self-quench, and then you'll have almost no fluorescence. Or we can mix a dye with a quencher. In both cases, we are expecting a very weak fluorescence or no fluorescence of the micelle. We can alternatively use a mixture of two dyes that can form a thread pair, or in the example that I would show you, we can label it with a dye that can form hexamers when in very close proximity. Now, the next step, we're going to introduce the enzyme that can degrade these amphiphiles. Now, the enzyme is not going to do anything to the dye itself. It's degrading the hydrophobic end groups from the other side of the polymer, but it makes the polymer to become hydrophilic, and therefore the polymer chains start to diffuse away from each other and leave the micellar structure. And now when the dyes are getting further away from each other, they can no longer interact, and we expect them to go back to the intrinsic spectral properties of the dye that we use for labeling. So to give you an idea about the synthesis, we start with the synthesis of the dendron. So we start with a branching unit that we prepare in the lab, having these two propargyl groups. Using a thioline click chemistry, we could convert each of these triple bonds into two branches containing already the aliphatic chain and the ester, that in this case will be cleavable by an esterase. Next, we can attach this linker for the labeling dye based on a lysine amino acid, giving us both the Boc protected amine and another new propargyl that would be later used for clicking the polymers. We deprotect the amine and introduce this aminocoumarin dye that we know that to give hexamers. And now we have this propargyl group that we can use to click with different polymers. And in this case, we chose to click it with either a polyethyloxazoline, which was functional with an azide on some chain end, polyethylene glycol, or polyacrylic acid. And the work that I'm showing is all the work of my student Gadi Slor, who is about to complete his PhD these days. And you can see already here in the photo the very nice spectral properties of the micelle that has this yellowish fluorescence compared to the hydrophilic or to the hydrolyzed, either the hydrophilic precursor or to the hydrolyzed version, which gives this very bright bluish fluorescence. Just to give you an idea about, again, the integrity of the structure. So you can see here the proton NMR. And again, due to the high symmetry of the dendron, you can really distinguish all peaks of all protons along the molecule, and integration works perfectly. You can see here the end groups, the methyl that sits at the end of the polymer, the coumarin, and really looks almost like an NMR of a small molecule. And if we go to the polymer, the PEG polymer that contains the nonyl end groups, you can see that nothing changed beside the peak here that simply contain more proton that sits in the slightly longer chains. We then obviously characterize the polymer. So as I said, you can characterize the polymers by HPLC. You can appreciate, so this is the peak of the dendron. You can appreciate the high purity of this amphiphile and their precise structure. And very nicely, again, due to this high precision, we can see that they all form micelles of roughly the same diameter. You can see here by the overlay of the DLS results. And you can see also here at the bottom that their critical micelle concentration are all very similar, roughly in the range of a few micromolar, with a slight decrease in CMC when you go from the hexyl to the nonyl-based hydrophobic end groups. Now, taking advantage of the fact that we have different fluorescence for the unimer and different fluorescence for the micelle, we did the following experiment to try and study their stability in presence of serum proteins. And what we did is we mixed our micelles with BSA that was labeled with Cy5. And the nice thing is that if the BSA with the Cy5 will be able to pull out polymers from the micelle for the unimer micelle equilibrium, then the unimers will interact with the BSA and would go back to their intrinsic fluorescence, which is around at 480 nanometers. However, if the BSA is interacting with the micelle, not with the unimer, then we'll have a threat going from the emission of the micelle at around 560, and we'll see the fluorescence of the Cy5, as you can see here, just a bit below 700 nanometers. And what you can see here are the results for the 3 different polymers, for the 6 different hybrids, with the 3 different hydrophilic polymers and the 2 different hydrophobic end groups. And we can see that as you go from the hexyl to the nonyl end group, even though the change in terms of molecular weight is very minor, we actually see a significant change in the stability of the micelles. We don't see the peak that come from the interaction of the unimer with the BSA, so showing that these micelles are way more stable and the exchanges of unimer to micelle is much slower. We could see in all of them some threat going between the micelles and the labeled BSA. And when you go to the polyacrylic acid, and the interaction with the unimer is so strong that there are almost no more micelles and all polymers become unimers. If we go to the acrylic acid with, again, slightly longer hydrophobic end groups, we can still see here the fluorescence that come from the micelle and a very nice signal of the threat, showing that there is interaction between the micelles and the labeled BSA. With this, we went into some cell experiment, and what you can see here is that we can either take the total fluorescence that we can observe by confocal, or at every given pixel, we can actually create a ratiometric image by dividing the wavelengths that correspond to the monomer and corresponding to the micelle. And we can see also here that if you have slightly less hydrophobic end groups, then the polymers seems to be to internalize faster, but they seem to internalize as unimers and not as micelles. The one with the longer hydrophobic end groups, both for the polyethyloxazoline and the PEG, seem to be going quite slowly into the cells, but we can see inside the cells both micelles and only at a later stage some unimer. And you can see that the polyacrylic acid is behaving in a very, very different way, showing very strong background fluorescence and accumulation on the cell membrane. And when you increase the hydrophobicity, again, you can see very strong accumulation. Everything is green, so all micelles outside. But when it goes into the cells, it seems to be going as unimer and not as micelles. The next step, we wanted to study the ability of these micelles to encapsulate drugs. And so we decided to use 2 commonly researched drugs. One is camptothecin, and the other one is paclitaxel. And I want to remind you all that the hydrophobic domains are exactly the same. So in one case, we have all, in blue, it's all hexyl, in red, it's all the nonyl group. And the fact that we're getting very different results, both for the different drugs, but also within each library, shows that the drugs really don't sit just as the core of the micelle, but are also being highly influenced and probably are being encapsulated also in the interface and within the shell of the micelle. So we can really, based on this high molecular precision and the fact that the micelles are very similar, can really gain very interesting insight about the ability to encapsulate different drugs. And the fact that eventually to tailor a carrier system to a specific disease, we'll probably have to tailor it also based on its interaction with a specific drug, and we'll not be able to generate this universal carrier that can carry any drug to any disease. So far, I think that what I wanted to show you is that we can use this dendritic hybrids to really fine-tune their structure and gain a lot of interesting insight about the mechanism of their enzymatic degradation and the response to different enzymes. But we actually, in a way, so far actually made the problem even worse, because it's clear that if you want to create some polymeric nanocarriers, you want to increase micellar stability to make sure that your polymer would not go into, be injected into the body and would immediately fall apart due to the high dilution or due to interaction with other proteins before it reaches the target. And so to increase micellar stability, what you have to do is increase hydrophobicity or increase molecular weight. However, as I've shown you in the first example, you pay very rapidly in your enzymatic responsiveness. So if the micellar you increase hydrophobicity to make the micelle more stable, you immediately and very rapidly going to pay in obtaining micelles that actually are not enzymatic responsiveness, cannot respond or cannot get hydrolyzed by the enzyme anymore. And here I want to go back into the multi-responsive in a manner that creating micelles that can respond to two different types of stimuli as part of our goal to overcome this challenge of the need to balance between stability and responsiveness. And in this last part, what we created are micelles that have a tile group right at the center where we used to have the labeling dye. And these tiles can go dimerization. And then you form amphiphiles that basically have exactly double the molecular weight, but have at the same time are keeping their hydrophilic to hydrophobic ratio. And what we found is that these micelles that based on the dimers are extremely stable to enzymatic degradation. Now, if you reduce them back to you still have micelles, but you'll be based on on the unimer, or not on dimeric amphiphiles, then this can rapidly exchange between the micelle and unimer and can get caught by the enzyme that's sitting outside of the micelle, degrading the hydrophobic end groups and allowing the micelles to disassemble. And let me show you the result, the structure and the result behind this. So you can see here the structures, all of them have hydrophobic end groups. Again, we have esters group that can be cleaved by an esterase, and we have the lysine-based linker decorated with a short extra linker to introduce the thiol here, or a methyl thioether SRR control group. You can see down here at the GPC that you start with a molecular weight of around 6.6 kilodalton, and if you oxidize it, it doubles the molecular weight. And if you add a reducing agent such as DTT, you can go back to the unimers. So we have a reversible system going from unimers to dimers upon oxidation, and then back to unimers upon reduction. Now, what you can see up here is the release of a fluorescent dye, a Nile red. Nile red is highly fluorescent when it's sitting inside the hydrophobic core of the micelle, but it becomes very weakly or non-fluorescent when it leaves and go into the more polar aqueous environment. So what you can see, if you take a micelle that is based on the dimer and you add the cleaving enzymes, nothing happened. The micelle is extremely stable and the Nile red is still sitting inside the core of the micelle. If we take the micelle based on the dimer and we add just a reducing agent, we see still high fluorescence because we haven't interrupted or we haven't changed the amphiphilicity of the amphiphiles. We simply move from dimers to unimers, but we still have the micelles and the Nile red can still sit there and be highly fluorescent. And it's only when we're adding both the reducing agent and the enzyme, we can see very nice decrease in fluorescence, indicating that the micelles are falling apart, being degraded by the enzyme and falling apart, and the Nile red is being forced to migrate into the more polar aqueous environment, and therefore its fluorescence is significantly decreasing. And we can see that we're seeing the exact same trend in kinetics if we use our control structure that have a methyl here, and therefore cannot go this dimerization. We're seeing the exact same release rate. And also, if we follow the degradation of the amplifier directly by HPLC, we can see also in this case that it's only when we have the dimer with the dimer-based micelle with the enzyme and reducing agent, we're getting this complete degradation as we follow by the HPLC, the exact same rate as we're seeing from the control structure, and there's a great correlation with the fluorescence result that you see up here with the HPLC, showing that it's indeed the reduction and going back to unimers, unimer-based micelle, and then the enzymatic degradation is that leading to the enzymatic disassembly. With this, I would like to conclude that I think that it's really I can say it's really paid off to do this extra synthetic work, even though I think we really managed to optimize the synthesis and we can make this a hybrid structure now on a multi-gram up to 20 gram scale of these amphiphiles with a reasonable cost and very high yield. And it's this really high molecular precision that allows us to dig better and understand what are the challenges and start providing some solution to solve them, hoping to see more and more enzyme-responsive polymeric nanocarriers in biomedical applications. With this, I would like to thank my group member, Karen and Paz, a lot of the funding agencies, collaborators from Tel Aviv University and abroad. Specifically here, I would like to thank Professor Lorenzo Albertazzi, who's the group doing the confocal microscopy, and Professor Richard Hugenboom, which together we worked on the polyethyloxazolin, and I think so is giving a talk tomorrow. And with this, I would like to thank you and be very happy to answer any questions. Thank you very much.
Okay.